This video is going to be about cell motility. Cell movement is dependent on cytoskeletal elements, in this case actin filaments and microtubules. There are two types of cell movement. Cells can neither crawl or swim. Here's an example of leukocytes homing in inflammation. We can see that this part is pulling this part, and this is causing the cell to crawl forward. This is an example of a cell that's swimming. In this case, it's a sperm cell. Cells crawl in inflammation, as I showed you in the previous example, in blood clotting, wound healing, and cancer spread. There are other examples, these are just the main ones. The first thing that happens is that at the leading edge, so this edge, actin filaments polymerize. This means that their extensions pull the cell forward. Microtubules then polymerize to stabilize this region. The myosin motors along the actin filaments contract, pulling the cell towards the newly extended front edge. This is how it moves forward. And this shows you how it depends on the actin filaments. The actin filaments are the ones that are dynamic and that polymerize and pull the cell forward. And how the microtubules are used to stabilize everything. And the myosin motors along the actin filaments pull it together. The direction of movement is dictated by chemical signals. So there are chemical signals that cause a response on the cell surface, which dictates direction of movement. Cells swim using flagellum and cilia. These are more characteristics of prokaryotic cells, although there are some eukaryotic cells which possess these. But their structure is different in eukaryotic and prokaryotic cells. Flagellum are outward projections of cell interior, and they contain cytoplasm on the inside and are enclosed by a plasma membrane. It can either be single or in pairs. Cilia are kind of like flagellum, but they're shorter and they're organized in rows, and the cell can have numerous cilia. They usually be in unison. Both these structures are organized in a 9+2 structure called axoneme. This means that there are 9 microtubule pairs, which are these around it. You can count 1, 2, 3, 4, 5, 6, 7, 8, 9. These are called doublets because they're in pairs, so double. And they surround 2 central microtubules. That's why it's called 9+2, because there are 9 around it and 2 in the middle. Pairs of microtubules are joined together through a linker protein called nexin, the central pair in the middle, and they move past each other using motor protein, which is called dynein. Flagellum or cilium undulates. They derive from this basal body, which is situated within the cell boundary. So this is as if it were the interior of the cell. There's this basal body, and the flagella derives from that.